Hello everyone, today we are from Group 6. We'll explain about De-Extinction, which is gene cloning in resurrection biology. So, this is our group members, and these are the contents that will be included in our presentation. Resurrection biology, or de-extinction, is the process of reviving extinct species by creating genetically identical copies from preserved somatic cells. The aim is to reverse plant and animal extinctions by generating new versions of lost species. Advances in selective breeding, genetics, and reproductive cloning have raised the possibility of bringing back extinct species through methods like cloning, genome editing, and backbreeding. However, while the extinction offers benefits such as restoring biodiversity and ecosystem functions that lost due to extinction, it also raises ethical concerns about genetic diversity. Now, we will look into the relevance of gene cloning in resurrection biology. The process of gene cloning plays a crucial role in resurrection biology by enabling the creation of genetically identical copies of an extinct species from preserved somatic cells. Cloning is the most widely proposed method for the potential restoration of an extinct species, as it allows for the recreation of an animal with the same genetic identity as the extinct species. This preservation of genetic material serves as the foundation for resurrection biology, allowing the reconstruction of the extinct species genome. The reconstruction of extinct genes through cloning could be used to restore genetic diversity in threatened species or subspecies, a fundamental effort in the restoration process. This effort can also contribute to the conservation of unique traits and evolutionary lineage that would otherwise be lost. By preserving the genetic heritage of extinct species, gene cloning facilitates the study of previously extinct animals and the potential discovery of cures to diseases. Other than that, by reintroducing resurrected species into their suitable habitats, gene cloning could significantly contribute to the revival of natural processes and facilitate the rebalancing of ecological communities. The reintroduction of resurrected species into suitable habitats can increase biodiversity and ecosystem resilience, contributing to the restoration of natural processes and the balance of ecological communities. Thus, cloning can be a pathway to revive lost biodiversity and restore ecosystem functions.
The general workflow of resurrection biology process involves a few steps. The first step is sample collection. Samples of bacteria or seeds from extinct species, such as horn, fur, or feather, are collected, which are then preserved for future use. The next step is genetic material retrieval. The nucleus from a preserved cell of the extinct species is extracted. Once the genetic material is extracted, it needs to be amplified using techniques such as polymerase chain reaction to obtain enough amounts of genetic material, such as DNA, for analysis. During DNA amplification, specific regions of DNA are chosen and replicated in large quantities. Sequence of extinct species DNA is compared to the sequence of a closely related living species to uncover genetic similarities and differences. The genetic material of the extinct species may need to be modified to make it compatible with the genetic material of a closely related species. This can be achieved through genome editing techniques such as CRISPR-Cas9, which allow for the precise modification of specific genes. Once the genetic material has been modified, it can be used to create a clone or hybridized version of the extinct species. The modified genetic material of the extinct species is inserted into an egg from a closely related species, and the egg is then implanted into a surrogate mother. The final stage of the de-extinction process involves rearing the clone or hybridized animals and then releasing them into the wild. This stage is crucial for the success of the de-extinction process, as it allows the animals to establish themselves in their natural habitat and contribute to the restoration of the ecosystem.
There is a few highlight key laboratory techniques in gene cloning in resurrection biology, including PCR. PCR allows the selective amplification of a target DNA region by using a DNA template, primers, DNA polymerase, and nucleotide building blocks. This results in the exponential replication of the target DNA, making it easier to work and analyze. There is a few steps in polymerase chain reaction, including designing primers, PCR amplification, gel electrophoresis, purification, and ligation. In designing primer, the first step is to design primer that will amplify the gene of interest. Then these primers are typically designed to include restriction enzyme that will allow the insertion of the gene into a vector. Then, PCR amplification, where the primer are then used to amplify the gene of the interest using PCR. This process involves the use of DNA polymerase enzyme to synthesize new strain of DNA from the template DNA. Gel electrophoresis. The PCR product is then separated using the gel electrophoresis, allowing the visualization and identification of the desired DNA sequence. The last one is to purification. The PCR product then is purified to remove any contaminants and to ensure that only desired gene is present. Then, moving on, the PCR product is then purified to remove any contaminants and to ensure that only desired gene is present. Then, the last one is ligation, where the purified gene is then inserted into the vector using the DNA ligase, which join the DNA fragment together. The extracted DNA is then sequenced using advanced technologies such as a high-throughput parallel pyro sequencing. This will allow for the establishment of the order of the DNA molecule for base or chemical building blocks. There is two steps in the sequencing method, which is first is assembly. The sequenced DNA is assembled using a complex algorithm to find overlapping stretches of the DNA produced by the sequencing machine. This can be done through the novo assembly or reference-guided assembly, where the high-quality genome sequence of a living relative is used as template to help stitch together small fragments into a full-length chromosome. Then the next step is where we analyze the DNA. The assembled DNA sequence are then analyzed to compare the DNA of the extinct species to the closely related existing species. This comparison allows for the identification of specific genes and the genetic variation that are unique to the extinct species. There is one more gene editing technology that can be used in resection biology to bring back extinct species, which is CRISPR-Cas9. The technology can be used to modify the DNA of a living organism to resemble that of the extinct species. Research can extract DNA from the preserved specimen of the extinct species and use CRISPR-Cas9 to edit the DNA of a closely related living species to match the extinct species. For example, scientists have used CRISPR-Cas9 to edit the DNA of elephant cell to resemble that of the woolly mammoth and extinct species. However, the use of CRISPR-Cas9 in resection biology is still in early stage, and there is many ethical and technical challenges that need to address before it can be used to bring back extinct species on a large scale. The CRISPR-Cas9 gene editing process can generally divide into three steps, which is recognition, cleavage, and repair. First, the design the sgRNA, where the process begins with the design of the single guide RNA that is complementary to the target DNA sequence. The sgRNA is designed to direct the Cas9 enzyme to the specific location on the DNA. Then, the CRISPR-Cas9 component, including the sgRNA and the Cas9 protein, need to be delivered into the target cell. This can be achieved through physical, chemical, or viral vector method. And the third step is recognition and cleavage, where once inside the cell, the sgRNA guides the Cas9 protein to the target DNA sequence. The Cas9 protein then creates a double-stranded break in the DNA at the target cell. The last one is to repair. The cell natural DNA repair machinery can be harnessed to introduce the desired genetic change at the site of the double-stranded break. This can result in the addition, removal, or alteration of specific DNA sequence.
Benefits of resurrection biology in medical applications. So theoretical in nature, resurrection biology has potential medical uses. The possibility for extinct species to reappear and contribute to biodiversity and ecosystem restoration is one of the main advantages. Keystone species might be reintroduced by this technique, improving ecological balance and possibly resolving problems like ecosystem degradation or overpopulations of particular species. Research on de-extinction may provide important new understandings of genetic variety and adaptation. It may be possible to identify distinctive characteristics or resilience elements by studying the genetic compositions of extinct species, which could improve present conservation efforts. This information might be essential for protecting already endangered species and habitats. Resurrection biology may contribute to improvements in regenerative medicine. Researching the processes that allow an extinct creature to resurrect could lead to advances in living things, tissue regeneration, and organ repair. Even while these potential advantages are fascinating, responsibly navigating the complicated field of resurrection biology requires rigorous regulation, ethical concern, and environmental impact evaluations.
Next is containment procedure. Firstly, biological safety cabinets. Use Class 2 or Class 3 BSCs for gene cloning procedures. Perform cloning work within these cabinets to provide an additional barrier between researchers and potential hazards. Secondly, decontamination protocols. Develop and adhere to strict decontamination procedures for equipment, work surfaces, and waste. Use effective disinfectants and autoclaving processes to eliminate potential contaminants. Thirdly, air filtration and ventilation. Install HEPA filters in the ventilation system to minimize the release of airborne particles. Regularly maintain and monitor ventilation system to ensure they comply with BSL standards. Waste management. Establish procedures for the safe collection, segregation, and disposal of biological waste. Autoclave or treat waste before disposal to eliminate any viable biological material. Lastly, biosafety level BSL determination. Conduct a thorough risk assessment to determine the appropriate BSL for the specific organism and materials involved in gene cloning related to resurrection biology. Consider the nature of the genetic modification and the potential risks associated with the resurrected or modified organism. Ethical consideration. First, practicality and efficiency. The expense and inefficiency of somatic cell nuclear transfer (SCNT) for resurrecting extinct species have raised questions about its practicality and feasibility. Altering natural history. The potential of cloning and other technologies to alter the course of natural history is a significant concern. Resurrected species may have difficulty adapting to current environmental conditions and producing viable offspring. Third, conservation efforts. Resurrected species would require conservation resources, which may be limited or lacking in some cases. This could lead to competition with endangered species that need protection. Animal welfare. Cloning and de-extinction raise concerns about animal welfare, as cloned individuals may suffer from unnecessary suffering. For example, the clone goat Dolly experienced health issues and died prematurely. Lastly, designing natural objects. Some argue that recreating and resurrecting species is creating artifacts that lack the integrity of natural objects, contributing to the domination of the natural world by humans, which is morally wrong.
For the last one is real-life example of resurrection biology. One real-life example of resurrection biology is the attempt to bring back the extinct passenger pigeon, which was driven to extinction in the early 20th century due to market hunting and habitat loss. The Revive and Restore project focuses on identifying mutations in the DNA that would cause a phenotypic difference between the extinct passenger pigeon and its closest living relative, the bent-tailed pigeon. In doing this, they can determine how to modify the DNA of the bent-tailed pigeon to change the traits to mimic the traits of the passenger pigeon, symbol of the potential applications of resurrection biology in conserving endangered species and restoring lost biodiversity. Now, we will look at a short video to summarize and enhancing our understanding of this topic.
The vast mammoth steppe of the Pleistocene era. Woolly mammoths, now long extinct, once roamed these northern landscapes in large herds, playing a critical role in the stability of their ecosystem. In the last half-century, however, these permafrost regions have begun to thaw at an alarming rate due to climate change and the loss of this keystone species. Over the last decade, humanity has made significant advances in understanding DNA and the field of genetics. Breakthrough genetic engineering technologies like CRISPR and others have made it possible to read, edit, and even write genomes. Combining these modern genetic engineering technologies with ancient mammoth DNA recovered from frozen specimens allows for the de-extinction of the woolly mammoth genome. Reintroducing the woolly mammoth to their former homes in the Arctic regions will help to bring balance back to the habitat and slow or even reverse the damage that climate change has done to our planet. Colossal. Restoring the past for a better future.
And these are the references, and with that, we thank you. Bye-bye.